Okay, RNA. So previously you've been introduced to a different type of nucleic acid, which is DNA, deoxyribonucleic acid. And again, throughout your entire journey of science within school, you've learned all about the importance of DNA, how you find a molecule within the nucleus, and as I just said there, how it holds this genetic code, okay? But what we're going to introduce you to now is that when we talk about gene expression and protein synthesis, we're talking about a different type of nucleic acid, okay? We're talking about RNA, ribose nucleic acid, okay? So when we talk about protein synthesis, and as we've mentioned previously, it involves two, so protein synthesis involves two important steps to go from the gene to the protein, okay? So transcription and translation involves three different types of RNA. And this video is going to take you through all three of them, okay? But before we do that, remember when we looked at in class the structure of a DNA nucleotide, okay? And how we spoke about, it's like my class, remember I always try and make the silly joke that people who told me years ago about when you look at a nucleotide, it looks a wee bit like a house with a satellite dish and a garden, or I think actually the boy said a swimming pool, but don't get a lot of swimming pools in Houston. So, but essentially, an RNA nucleotide looks the same, but there's different components, hence why we cannot no longer call it DNA, okay? So it still has a phosphate group attached at the end, okay? And remember, it's the same as it was before, okay? So we still have our same number of carbons. So 1, 2, 3, 4, fifth carbon, okay? However, this is a different sugar, okay? So it's no longer deoxyribose sugar. It is now just known as ribose sugar, okay? And this is still our base. But the difference being that with DNA you have your adenine, thymine, guanine and cytosine, okay? But what you have here is you still have adenine, but you no longer have thymine. There's a change in a base. So you now have a base called uracil, okay? And then you still have guanine and cytosine. And it's the same as before. Complementary pair, guanine and cytosine, and adenine and uracil is still your complementary pair. So this whole nucleotide, this whole diagram is an RNA nucleotide, okay? And it would be the same as before, that you have all the nucleotides, so the two strands of DNA is two strands of a complete order of nucleotides joined together by strong hydrogen bonds, and that's what's making this what we'll call the sugar phosphate backbone of the molecule. However, again, the difference between DNA and RNA is that RNA technically, although we still have nucleotides, okay, but they're RNA nucleotides because they've got different components. The next, the kind of, in fact, it's not the next, the first type of RNA we're going to look at is messenger RNA, which is no longer a double-stranded molecule like DNA, it is a single-stranded molecule, okay? So, messenger RNA, okay, so our first type of RNA that we're going to look at, okay, is messenger RNA, okay? So what you see abbreviated is mRNA, okay? And essentially what this is this whole strand of these nucleotides. Okay, so again, as I said, same as DNA, they're all joined together by strong hydrogen bonds, but the difference being is that it's single-stranded. There is no two strands to make the full molecule. To make the full molecule of DNA, you need two strands, whereas messenger RNA is single-stranded. Okay, so again, phosphate, ribose sugar, and a base, all joined together. Okay, but again, remember we have adenine as our base, uracil now, no longer thymine, guanine, and cytosine. Okay, so what we'll do later on in one of the next videos is we'll talk about the formation of this molecule, what exactly happens, because we need our DNA, we need our code that's on our DNA molecule to therefore make this single-stranded messenger RNA. Okay, and the messenger RNA molecule is in relation to transcription, the first stage in relation to protein synthesis. Okay, and that's when we talk about, again, we'll go over this in a later video, so transcription of a copy of the DNA code in the nucleus. Okay, because we don't want to, when we're making a protein, we're not wanting to go into the DNA molecule and remove that section, because then it wouldn't be a functioning working molecule anymore. So what we need to do is we need to have this double-stranded molecule open up for these RNA nucleotides to go in at the exact area. So we call it a kind of replication bubble, you know, where we're going to replicate that code, take a copy of that code to form this molecule to therefore go on to the next part of protein synthesis. Later on we'll talk about translation. Okay, so this is an important part of protein synthesis. Okay, so if we go into it a wee bit further, okay, you'll understand later on why I'm using a square jotter, and it's more just to make sure that I can kind of do the diagrams properly in relation to lines. Okay, so I'm going to kind of demonstrate that just now. So when we talk about creating a copy of this code, what we want to do, so if you can imagine it here, so here I'm going to draw DNA, right? Okay, so here's a strand of DNA and I want to work this out properly. So I have A, T, G, C. I'm just going to, for ease, I'm just going to keep on repeating them. C, G, A. Okay. So hopefully you recognise that this is one strand of a DNA molecule. Okay, and remember, DNA found in the nucleus. Okay, codes for proteins. Okay, this is what we're looking for. Remember, the ultimate goal is to make that protein. So here we've had to say there's another strand here, and it's opened up, and this is our opened area. This is the gene that we require to make the protein. Okay, so what's going to happen is free RNA nucleotides, and again we'll go into greater detail in a later video, okay, are going to come along and they're going to match, i.e. they're complementary pairs. Okay, so if we've got A, T, G, then that means a nucleotide, an RNA nucleotide, okay, with a uracil base, because remember it's not thymine anymore, will come along, okay, and it's going to pair up with its complementary partner. Okay, so we're going to have U for that. That would be A, that would be C, that would be G. So again, different, not thymine, it's going to be uracil, still adenine, guanine, cytosine, and again, uracil. So remember, these are our different ones, these in relation to RNA. Okay, so now I can say, well, there you go. Here is my messenger RNA strand starting to be produced. Okay, so this is going to be my messenger RNA. And the thing is as well, we need to remember that 1, 2, 3, okay, so a triplet of nucleotides is called a codon. And this is in relation to messenger RNA, which will make sense later on when we talk about transcription. Okay, so again, 1, 2, 3, here is my codon. 1, 2, 3, here is my other codon. Okay, so that's DNA, messenger RNA. Okay, so we can say it's a copy of DNA code, and again, synthesized. Okay, so remember, synthesis just means to make something. Okay, so it's synthesized in the nucleus, because that's where the DNA is, and that's where the code is, that's what we want to copy. Okay, and moves to the nucleus. Moves to the ribosome. Okay, and again, we'll come back to that part later on. So the essence of messenger RNA is it makes a copy of this specific gene. So this is what we want. So gene for protein. Okay. And this is a copy of gene for protein. Okay. And this messenger RNA is going to go and travel from the nucleus, okay, to the ribosome. All right, so messenger RNA. All right, okay. So the next one that we're going to talk about is transfer RNA. Now, as I said, there's three types of RNA. Okay, so messenger RNA, and now we've got transfer RNA. And transfer RNA is the one that it's a wee bit more difficult to draw, but it still has a very important role within protein synthesis. Okay, so transfer RNA. And it's represented as tRNA. Okay. Not as, now it's still, and this is where a lot of people get a bit confused with some exam questions, it's still a single-stranded molecule. It's just, its structure makes it look like it is double-stranded, but it's not, it's a single-stranded molecule. Okay. So, in saying that, it's folded, and it's these folds that gives a pupil an impression that it's double-stranded. Okay, so it's a folded structure, and it happens because of its complementary pairing. Okay, so folded structure due to its complementary, see if I can spell complementary base pairing. Okay, it's still made up of RNA nucleotides. Okay, as I said, real important to remember, single-stranded, okay, and it has two very specific areas, okay, so it has an attachment site, okay, that carries a specific, remember this term from enzymes, okay, carries a specific amino acid. So there's different types of transfer RNA molecules. Although the structure's all the same, they carry different amino acids, okay? And then it has, at the bottom of it, an anticodon. And remember, when we look at the codons here in messenger RNA, the transfer RNA, okay, has an anticodon, and this is where these codons and anticodons are going to bind together. Okay, so when we look at the structure, again, not an artist, so I'll try my best. So the structure is almost kind of, as I say, largely related because of these individual folds. Okay, so essentially what's going to happen is this is all one strand. Okay, but because we have, so if I draw these on, so I'm trying to give you examples of why there's folds there. Okay, so for instance, let's see, yeah, we'll keep on going inside. So technically what I'm drawing is the bases. Okay, so that could be A, U, G, that could be C, G, U. Now remember, if this is A, U, G, then because these are the complementary, so these are the wee weak hydrogen bonds, okay? Okay, so because these are complementary, the next base must be U. That would be A. If that's G, then that would be cytosine, and so on and so forth. Okay, so again, that's why it's folded, because these, so naturally, these nucleotides want to pair with each other, which therefore creates a pool. Again, more of a kind of chemistry or physics kind of explanation. A physics teacher to kind of explained about this kind of natural pull between the nucleotides, the individual bases. Okay, but it does. So that's what creates this structure. And then as you've got these folds, what you also have, as I said before, is at the bottom you have this section here that carries individual, and they're not, I'm only making it large just for the kind of diagrams purpose, but they're the same as this. Okay, but at this bottom here we have what we class as our anticodon. And again, that could be A, U, G. Okay, so at the bottom we have our anticodon, and then at the top, this is where we have our attachment site. And what we mean by an attachment site is we have a specific type of amino acid. Okay, and again, remember, you guys don't need to remember or be able to recall the names of amino acids. You might see them in exam questions, but they're just providing you that information for the question. There's no need for you to know them or recall them. But the importance is that you know that there is amino acids. So that amino acid belongs to this transfer RNA molecule, and it's to do with these anticodons here. So if that has AUG, then say that's the amino acid leucine, whereas another transfer RNA may have GCA, which means that's a different type of amino acid. That's what we mean by different types of transfer RNA molecules, just all to do with these codes, this anticodon in relation to this amino acid. Okay, and essentially what's going to happen is this individual molecule, transfer RNA, with an anticodon is going to bind onto the codon of this molecule here. Okay, so we have 3 different types of RNA. First one here, okay, so here we go here, messenger RNA, okay. We now have transfer RNA, okay. I think what I'm going to do is I'm just going to put it in the bottom of this one here. And our final type of RNA is ribosomal RNA. And this is the one that you just need to know the terminology. That's all it is. You just need to know that there's 3 different types of RNA, and one of them is rRNA. Okay? And essentially all this is the RNA that makes up the ribosome, where the protein is finally produced, where messenger RNA travels to, where it meets transfer RNA, and the ribosome will move along and produce the protein. Okay? To give you a wee bit of greater kind of detail, is the fact that the ribosome is a large organelle within the cell. Okay, it's got a slightly smaller, so it's got a large subunit, and it's a small subunit. if you guys want to go to university and do biology further, you'll learn lots about these different units. And then essentially what's going to happen is our messenger RNA is going to move along. Okay, or the ribosome moves along the messenger RNA, and out comes the protein. Okay, so 3 types of RNA: messenger RNA, transfer RNA, and ribosomal RNA.